Good morning, everyone. My name is Nora Ventosa. I love ICMAB, so very much. So I am very pleased to share this session during this 40th anniversary of our institute. And it is my pleasure to introduce you Carmen Freire. She is a coordinating researcher at CICECO from Aveiro Institute of Materials. Carmen is industrial chemist and she did the PhD at the University of Aveiro. She was postdoc at the Institut Politècnic de Grenoble, and she is worldwide recognized for her research and contributions in the field of the development and characterization of natural polymer-based materials. Her expertise is recognized by being member of several of the board of several major journals in green chemistry and material science, and we thank her very much for being member of our ICMAB Scientific Advisory Board. So, Carmen, the floor is yours.
Thank you so much. Good morning to all. First of all, it's really a pleasure and an honor to be here, to be part of the Scientific Advisory Board of ICMAB, and particularly to be here today celebrating your 50th anniversary, but particularly celebrating the excellence of the science that you are doing here. So thank you so much for the invitation to be here today. So we will change completely the subject of the talks, and I would like to share with you some of the work that we are doing at our group on the field of novel biopolymeric materials. Here I will focus on materials for healthcare applications, but we work also in other areas, as I will show you. So just for those that do not know me, I'm coming from Portugal, so from the Department of Chemistry of the University of Aveiro, as introduced it. So Aveiro is a very, very pleasant city in the north coast of Portugal, and our Department of Chemistry is located in these two buildings, here in a very pleasant environment. Our university is very young. In fact, we are celebrating this year, the Department of Chemistry is celebrating the 50th anniversary. So we are very young, but already a very important university in Portugal in several areas, including in the field of chemistry. In terms of research, we are part of CICECO. CICECO is the Aveiro Institute of Materials and is the largest institute in Portugal in the field of material science and engineering. So we are organized in different transversal research lines, and different groups. Our research team, the Biopol for Fun, is a research team that belongs to Line 3, Sustainability, and Group 4, Renewable Materials and Circular Economy. And we are working essentially towards the innovation in the field of functional biopolymeric materials, but also bioactive compounds. We work also a lot with low molecular weight compounds. Here you can find some numbers about the composition of our group. This is always changing, but just to have an idea of the dimension of our research team. As I already mentioned, our goal, our vision is to take advantages of the diversity of natural polymers that you can obtain from different sources, and together with the use of different processing technologies, we can produce innovative, sustainable solutions for different applications. So this is the focus of our research. We work essentially on the design and characterization of the materials. Obviously, the selection of the natural polymer, the processing technology strongly depends on the application. And of course, we have been working in different fields with different materials for different applications. Today, I will focus only on some examples of materials for healthcare application. And I select these three. It was not easy, But I decided to select these three because bacterial cellulose-based materials are one of the oldest research areas of our group. So we work for a long time in this field. And these two more recent topics, biopolymeric microneedles and hydrogels for 3D bioprinting application that are more recent, but we are working a lot on these topics nowadays. Starting with bacterial cellulose, I think I don't need to go into much detail here. We have a strong group working here in ICMAB in bacterial cellulose. So we implemented several years ago, I can say about 20 years ago, the production of this cellulosic substrate in our lab. We work essentially with membranes. Okay, I don't know if this is working. It's here. It's here. We work essentially with membranes, but also in some works we produce beads, depending, of course, on the cultivation conditions. But in any case, the most important aspect is that this cellulosic substrate is characterized by this peculiar 3D nanofibrillar structure. And this translates into a set of unique properties that we cannot find, for example, in plant cellulose. And it has been considered and exploited as a unique nanostructured bio-based porous materials. And in fact, is this peculiar porosity and porous structure that allowed us to produce different materials, for example, by the simple diffusion of different molecules inside this 3D porous structure. And depending on the molecule that we use, of course, we can tailor and adjust the properties and the application that we are looking for. And in this area, we started several years ago working on the development of membranes for dermal drug delivery. We tested several different drugs following a very simple approach, just by place the wet membranes in a solution containing the drug and also glycerol to guarantee a good malleability, and also glycerol is used as a humectant for skin formulations. And then following oven drying, we are able to obtain membranes that are very homogeneous and that easily adhere to different regions of the body. And this is very good because depending, of course, on the drugs, we can have the formation of agglomerates, but in most cases, this was not the case. So we are able to obtain a good dispersion of the drugs inside this network. That is an important aspect. And even in most of the cases that I refer, the permeation profiles that we evaluate using ex vivo human skill and these Franz cell diffusion cells are very comparable to commercial formulations that we use to compare the performance of our membranes. And in fact, they can be used as alternatives to these formulations because, in fact, we have very comparable profiles. We realize that the drug permeation rate depends essentially on the nature, the chemical nature of the molecules and the interactions that are established between the molecules and the fibers. So if we want to go into a target release or even a more controlled release, we need to go through more complex systems. And in particularly, we have been investigating pH-sensitive systems and multi-layered patches to try to achieve these objectives. In this work, we prepared a pH-sensitive membrane by the in situ polymerization of a monomer inside the membrane. So in this case, we incorporate the monomer, we polymerize it, and then we obtain a nanocomposite, as we can see here. And after that, we include a drug, in this case, diclofenac, that was used as a model drug. We also prepared beads aiming the oral administration, but in any case, these systems present high water uptake, good thermal and mechanical properties, are non-cytotoxic toward human cells, but most important, we are able to obtain a pH-dependent release. That was what we intend, for example, particularly for oral administration, to avoid the effects of the first passage in the stomach. In this study, we decided to prepare these biopolymeric multi-layer systems, and we achieved this by the deposition of alternate layers of chitosan and alginate, and this allowed us to prolong the release of the drug depending on the number of layers. So we can adjust the kinetics of the release of the drug depending on the number of layers, and this could be important depending on the clinic application that we want and that we desire. Obviously, depending on the polymers that we select and that we use in this system, we can incorporate additional properties. And in this case, since we use chitosan, these membranes also present antibacterial properties. They are, of course, non-cytotoxic, and the presence of the drug that, in this case, I think I forgot to say, was dexpanthenol, a common drug used in skin formulations for skin regeneration, improved the wound closure in this in vitro model. More recently, we decided to prepare multi-layered systems, but using only bacterial cellulose. So we only use these bacterial cellulose membranes, and here the objective was to produce a multifunctional patch that can be used on the treatment of herpes labialis. And we achieved this by using acyclovir, that is a common drug used for the treatment of this disease, and also hyaluronic acid, that is a biopolymer widely used for skin regeneration. And we are able to get other important properties. How we achieve these materials? First, we prepare bacterial cellulose membranes with different compositions, and then we assemble them in different morphologies in order to, for example, divide the amount of drug into the different layers, and also to guarantee that hyaluronic acid was always in the layer that will be in contact with the skin. And these different morphologies allowed us to adjust the release of the drug depending on this structure. In this case, in all these examples that I gave, we are only using one molecule, one drug, but we can also consider more complex mixtures. As in this study, this was a study that was carried out in collaboration with the pulp and paper industry in Portugal. It was a big project that had the objective of innovate in the area of products and technologies from eucalyptus. Eucalyptus is the most used tree in Portugal, but also Spain, to produce pulp and paper. And this project has a main objective of implement these bioeconomy concepts, circular economy concepts in this industry. And one work was centered on the use of sugars obtained from the deconstruction or residues, and also to produce bacterial cellulose, and also extracts from leaf residues as bioactive compounds. And in fact, we succeeded, we produced these membranes, very homogeneous, with interesting properties for cosmetic applications, and these result in a paper and a patent that was shared between the university and the company. And nowadays, they are scaling up the production of the extract, because the patent encompasses the production of the extract and of the membrane. So nowadays, they are scaling up the production of the extract, the extraction process, and we hope in the future we will be able also to go into the next step in the fabrication of these masks. In all these cases, we are talking about low molecular weight compounds, molecules, drugs. We can also incorporate polymers in following a different methodology. Previously, we were talking about the post-synthesis approach. So we produce the membranes, we purify, and then we incorporate, we change, we modify the membranes. Here, we took advantage of the biosynthetic process. And in this case, we use two different bacteria. So we co-cultivate different microorganisms to produce a composite. One of the microorganisms produce bacteriocellulose, the other hyaluronic acid. And at the end, we have a nanocomposite with interesting properties. In this case, we preliminarily evaluate the application in wound healing. And this was done in collaboration with an Italian university, a group from the microbiology field. And it was very interesting to really achieve the production following this biotechnological process. So without chemical modifications, without additional steps. Now moving to the biopolymeric microneedles. These are very small devices composed of micron-sized needles, and these devices combine the properties of the hypodermic needles and those of the transdermal patches. So these microneedles are able to pitch the stratocorneum and guarantee that we can administrate different drugs. That is the main problem of the conventional transdermal patches, because this stratocorneum is a barrier for most of the drugs. But since they not reach the nervous terminations, they are painful. So they can be used in different applications, fabricated following different approach, and have been explored in different fields in the later years. In our group, we are using the micromolding approach, and we have been fabricating different types of biopolymeric microneedles. The first study was a more conceptual study. Our objective here was to explore the use of these polysaccharide pullulane for the first time in the fabrication of microneedles. At that time, we had some experience with these biopolymers, for example, for films, because it's a highly filmogenic polysaccharide, but had never been used in this particular area. So this study involves, first of all, the optimization of the concentration of the solution, the adjustment of the viscosity of the solution to guarantee that we were able to fill the cavities of the molds. And then, of course, all the characterization to confirm that we are able to really perforate the stratocorneas and reach the desired skin layers. And in this case, we test the release of the incorporation and release of insulin, for example, for the application, the treatment of diabetes. Even this work was more conceptual in terms of the valorization of this microbial polysaccharide. More recently, we still continue to use pullulane, but now we are more focused on the application. And here the objective was the administration of an anesthetic. Anesthetic procedures are required in several clinical procedures, and sometimes they are very complicated because most people do not like to use needles and so on. And here we try to explore this approach to administrate lidocaine. Apart from all the steps regarding characterization, evaluation of safety, we implement the in vitro methodology to test the efficacy of these microneedles. We did this in collaboration with the biomedical department of the university, and we used a neural cell line as a 2D model, and we were using a fluorescent probe that was sensitive to sodium. Even it is known that lidocaine influences the calcium channels, it also has an influence in sodium. So we were able, following this approach, to confirm that in fact we are able to release lidocaine and to have the same effect as a solution contained the same amount of lidocaine. It was a very interesting collaborative work with the colleagues from the medical department. And this work is a combination of microneedles with bacterial cellulose. It was work that we performed some years ago, but it was very interesting because we thought, why not use bacterial cellulose as a back layer of the microneedles, first to reinforce the needles, but at the same time use this membrane to deliver another molecule and complement the effect of the needles. In this case, we use hyaluronic acid needles and this back layer loaded with rutin. Rutin is a phenolic compound also used in these healthcare applications. And this study was very interesting because we're able to do some trials with human volunteers to check the safety, but also the acceptance of this technology, because we made a questionnaire where they answer if they feel some pain, how they accept or not. So it was a really nice study. And with this approach, we are able not only to have the effect of hyaluronic, but also to introduce other properties as antioxidant properties and so on, based on the use of this back layer. And nowadays, we are using this approach to design living biomaterials. It's a project of a junior research of our group that is working toward this new class of biomaterials that incorporate living components. And finally, I still have some time, I think, the design of hydrogel bioinks for 3D bioprinting. As certainly you all know, bioprinting is the biological branch of 3D printing. So in this case, the material that is printed, that is deposited by the printers, contains living cells. And this allowed us to create 3D constructs that contain the living cells and can mimic living tissues. And where can you use this type of materials? In tissue engineering, of course, also as models to study diseases. It has been widely used in cancer research and also to test new drugs, to avoid the use of, for example, animal models in earlier stages of research. So this is now an important area in the biomedical field. Our work is essential on the design of biowings. So the success of the 3D bioprinting process depends on the printing technology, but also on the availability of suitable biowings. And a suitable biowings needs to fulfill several requirements. We are working with living materials, so we need to have biocompatible materials. They need to biomimic the living tissues. They need to be printable, present high printability to guarantee that we obtain stable constructs and, of course, suitable structural mechanical properties. And this set of properties are needed to guarantee that we create a similar environment to that of the extracellular matrix, and therefore guarantee that cells are viable and they can proliferate in this environment and create these living tissues that we want. There are different types of bioinks that we can use, but hydrogel stands out in this field, and particularly natural-based hydrogels. And in this realm, polysaccharide hydrogels have gained considerable attention, as we can see in this graphic, because we have a huge diversity of chemical structures that allowed us to have different functional groups, to adjust the chemistry involved, and in particularly because polysaccharides, most of them, can be cross-linked in mild conditions. That is important because we are talking about living cells. But not everything is perfect. Polysaccharide-based hydrogels present some limitation in terms of mechanical performance, and most of them lack active cues for cell adhesion. So how can we overcome? There are several strategies, and one in particular regarding the mechanical properties is the design of nanocomposites, is the addition of nanofillers into the hydrogel matrix in order to improve the rheology, the mechanical performance, and so on. We can use, there are in the literature several works using different nanostructures, carbon nanostructures, metallic, and natural nanofibers. Of course, we work with natural polymers. We have essentially using natural nanofibers, nanocellulose fibers, and protein amyloid nanofibrils. I will talk a little bit about this. Here, regarding nanocellulose, we are working essentially with nanofibrillated cellulose that is obtained by mechanical and enzymatic treatment of plant fibers. I will not go into detail, but you see that we have these isolated nanofibers, and these nanocellulose are essentially used as reinforcement to improve the mechanical performance and rheological properties. In this area, our goal was to test new formulation, to exploit polysaccharides that alone are not able to be printed. And for example, as in this study, we use gel and gum. The gel and gum hydrogels are very fragile. So when we incorporate nanocellulose, we're able to increase the printability, to obtain 3D constructs, and to have a high cell viability after 7 days post-bioprinting, here using skin cells. So here, this work focused on this new formulation and to use another polysaccharide and enlarge the diversity of available bioinks. The next work, it was as a different rationale. We work a lot with cellulose. It's one of the polymers that we most used. And nanocellulose is cellulose is essentially used in the form of nanocellulose as a reinforcement, not as a mattress. So here, our objective was to design what we call a all cellulose bioink, an ink composed only of cellulose. And we were able to succeed using carboxymethyl cellulose, but it was not easy because we needed to optimize the molecular weight, the composition in order to reach these results. Complicated structures, different structures, but in particularly high cell viability. We are able to adjust the mechanical properties, the rheology of the inks in order to meet the requirements of different cell lines here, keratinocytes and melanoma cell lines. So you are able, the chondrocytes, if I'm not wrong. So we are able to adjust the properties and select different cell lines. This is important. We need to go into or to meet the mechanical properties of the tissues because cells need to find an environment that is similar to the native one. We are not only using nanofibrillated cellulose. In this study, it was a completely different concept, and I really enjoyed this work. This work intended not only to use cellulose structures to reinforce the inks, but also to incorporate other properties. And we achieved this by using cellulose-based particles that were loaded with a model drug, a model compound, and then these parts were incorporated into the ink formulation. And this allowed us to produce a 3D construct that, apart from being living construct with cells, also has the ability to release a drug. And this was very interesting because, in fact, we are also able to control the degradation of the construct. That is also important when we are, for example, talking about tissue regeneration, because it's important that the material degrades in the same rate as the new tissue is formed. And also you are able to produce very nice structures with drug-releasing ability. And you can see here the particles embedded in the matrices. Protein amyloid fibrils. This is a quite emergent class of bio-based nanofibers. Probably you have heard about this family of nanofibrils because they have been for a long time only associated with some pathological disorders as Alzheimer's. because this disease has associated formation of these amyloid fibrils in the human body. But it is now known that these fibrils also play important roles in the human body and also in other living organisms as microorganisms. And these amyloids are called functional amyloids. And because of this, and the fact that now is known that almost all proteins and polypeptides can be fibrillated in vitro, they are gaining much attention. In our group, we developed a very interesting approach to fibrillate lysozyme from egg white that allowed us to reduce the fibrillation time from days to 2 hours. Is the methodology that you continue to use, and we are essentially use lysozyme, but now we are trying other protein sources. This aspect that we can produce in the laboratory, they have unique mechanical properties, but most important and quite different from cellulose. Cellulose probably have better mechanical properties, but is quite inert. And protein amyloid fibrils, they, in most cases, maintain the biological functions of the native proteins. And this is quite interesting and has been widely explored on the biomedical field, but also in other areas. Because so these fibers can have a dual function, reinforcement and bioactivity. In the 3D bioprinting field, we were one of the groups that pioneered the use of these fibrils in 3D bioprinting. This was a very simple and preliminary study where we just confirmed that we can use them. And apart from improving and modulate the rheological and mechanical properties, we also improve the biological properties. In this case, we saw an improvement in the cell viability. And after this very conceptual work, we have been using these fibers not only in bioinks, but also in other materials. And here, in this very recent work, we explore another feature of these protein nanofibrils. They can be used as templates for the synthesis of metallic nanoparticles. And here we use this approach to prepare hybrids of protein nanofibers and gold nanoparticles that were used as additives in an hydrogel matrix. And this was done to produce conductive bioinks for the bioprinting of cardiac cells. That is one of the properties relevant for this area. And apart from this, these bioinks present antioxidant properties. Probably this was the best bioink that we developed until now. We are able to produce quite different structures, and we were able to print at least 72 layers, keeping the stability and the structure of the construct. 2 minutes. I have only 2 more examples and I'm done. Okay. Just to finish. In this work, we decide to combine nanocellulose with protein nanofibers because, in fact, cellulose has a better performance in terms of rheology, but lacks biological properties. So with this, we are able to really improve the properties of a pectin hydrogel, and we will really succeed in terms of the printing of different structures. And here, we started exploring more the potential application of these living tissues. We started using this as a 3D platform to test drugs. Here we print tumoral cells, melanoma cells, and we test a model drug just to see if we can observe the impact of this drug in this more complex model. And in fact, we were able to do it. So this was a first study. Now we are producing more complex structures that try to mimic better the structure, the structure of skin, because here we only use one cell, and we are now producing more complex models that better mimic the native tissue. But this was interesting in terms of the concept. And finally, the last example. The objective is to print living systems, but to use them. And here we use these 3D printed structures to study the efficiency of a new microneedle system that we developed. Here we incorporated antitumoral polysaccharide, and we wanted to go further and confirm in a more complex system that, in fact, our microneedles were efficient. We first carried out 2D experiments with the cell cultures, but here, in fact, we moved and we saw that these systems are still effective in a more complex system. And so here we are joining our expertise in the different fields, and we are collaborating with other groups in testing their materials. So this is really the last one. And I don't have a conclusion. I just want to, I hope that with all these examples, I had demonstrate and show to you that natural polymers are really versatile, and we can design completely different materials with different functionalities following green strategies. So here, our objective is to have a contribution to these sustainable solutions with a lower environmental impact. And at the other hand, we design efficient materials that can also contribute to more efficient healthcare treatments and well-being in general. So thank you so much to you. I leave you with these beautiful images of Aveiro. So you are all welcome to Aveiro, to the university. And I want to thank, of course, everyone involved in these studies, to the university, the financial support, to you, and congratulations, ICMAB, for your 40 years. Thank you so much.
Thank you. Thank you very much, Carmen, for this nice talk on natural products for health applications. I think that we have time for a couple of questions. So the floor is open for questions to Carmen. Yes, there is one.
Hi, thank you very much for the interesting talk. I'm one of the big fans for the bacterial cellulose as well. Yeah. So as we all love bacterial cellulose, I have one, not a question, maybe your suggestion, how you see the potential for the industry scale up, because we do know the yield is quite low. So I would like to know, I would like to put this material into the industry, more application, but due to this limitation, so I would like to hear your suggestions. Thank you.
That is a question that I made to me several times. This is, for me, a unique material, but still we don't have too much industrial application. And I remember that I was in the first edition of the conference dedicated to bacterial cellulose in the US, and I heard a talk of a scientist that received millions to put bacterial cellulose into the market, but he did not succeed. I think there are several reasons, and particularly the application in the biomedical field is not easy because the medical sector, I think, it still needs more information, is not open sometimes for these new solutions, and it requires several regulations, and I think it's complicated, but I think we need to continue and create knowledge and prove that it is a good material. So that's why I continue to work with bacterial cellulose. I started about 20 years ago, and I still work in, and I'm still fascinated, but it's not easy to really go to the market. But there are already some products with small companies, some materials for wound healing, and so on. But still, I think it's far away from the potential of this material. I hope that in the next years, all of us can contribute and lead this material to the market. I hope so, but yes, it's not easy.
Thank you. Time for another question. Yes.
Thank you very much for this nice and impressive presentation. I have a question regarding the first topic that you have presented about the multi-layer polymer stacking.
The one with the chitosan and alginate?
Yes. How there is any effect about the polydispersity or molecular weight of the polymers that you are combining? So they have to be similar, or you can combine also.
I don't remember the specific molecular weight of the different components, the different polymers. Of course, the molecular weight have an impact, particularly on the viscosity, that is important for the homogeneous deposition of the layers. And we optimize that. I don't remember if they are similar or not, because they are different polymers, and they have, for each polymer, we have a different impact. But yes, the molecular weight has an important role here, and we need to have solutions with the appropriate viscosity for the homogeneous application of the layers, yes.
Okay, thank you very much. And please join me again to thank Carmen for the briefing. So we continue this session with a presentation from Juan Pellico. Juan Pellico is a Ramón y Cajal researcher from our institute, from the Institute of Material Science here in Barcelona. And he obtained his PhD in chemistry from the Universitat Autònoma de Madrid. He then performed several postdocs, the last one at the King's College Research in London. His research is focused on the use of nanomedicine for medical imaging. And he was recently awarded with an ERC Consolidator grant. So we are very proud of you, Juan. So please.
Thank you very much, Nora. And yeah, I had prepared a brief introduction, but Nora already made it. So that's absolutely fantastic. So I got straight to the point. I'm going to talk about the work we are doing in developing nanomaterials for medical imaging. As you may know, medical imaging are really powerful techniques. So we can have functional and anatomical information with very high precision. And today I'm going to talk to you about I'm going to give you some examples about 3 different imaging techniques. 2 nuclear imaging techniques that require radio-labeled contrast agents, and 1 that is magnetic resonance imaging that usually requires, depending on the case, a magnetic contrast agents. And that's the limitation I want to point out. So the current contrast agents have many limitations due to them based on small molecules. These small molecules have rapid pharmacokinetics. They are eliminated very quick from the body. And that limits the amount of functionalities we can do with our imaging techniques. That also limits if we can or we want to target a specific site with a small molecule is usually quite difficult. However, nanomaterials can overcome these limitations. We can control potentially much better the pharmacokinetics, and we can also perform better targeting, as we will see in the next couple of examples. So let's see some examples about the pharmacokinetics. Here I have an image of a clinically relevant contrast agent. This is MagneVist. It provides a positive contrast in the tissue where it's accumulated. That means a brightening in the tissue where it's accumulated. Maybe if I don't point out where to look, it might be difficult for you to see whether we have contrast or not. However, if we do the same with a nanoparticle with the same positive capabilities, but a long circulating particle, here I don't need to tell you where to look. You can clearly see the difference between the image without contrast and with contrast. And this is very important for our clinicians. Another important application that I found very, very interesting is the control of the pharmacokinetics to radionuclides that we cannot control now in the clinics. This is the case of thallium-201. Thallium-201 is a wonderful radiotherapeutic isotope, or at least it has wonderful radiotherapeutic properties. But we cannot use it just because one main reason. We don't have any ligand to coordinate thallium to deliver it to a specific place or to control the pharmacokinetics inside the body. In this case, what we did was to use Prussian blue nanoparticles. So essentially, we study, this is working, yeah. We study the inclusion mechanisms of thallium into Prussian blue nanoparticles, and most importantly, we demonstrated that after the administration of our Prussian blue nanoparticles, the thallium-201 is much more longer inside the tumor than the free thallium. So we can boost the radiotherapeutic potential of this radioisotope just by using nanomaterials. So I think these are good examples about the control of pharmacokinetics that we have with nanomaterials. I'm going to show you only one example about targeting. I think this is also like a nice example. We are targeting here atherosclerosis. Atherosclerosis is a difficult process to study in cardiovascular diseases. It's done by many different things. In this case, what we wanted to study is the formation of microcalcifications. Okay, so for that, what we did is to develop an iron oxide nanoparticle, okay, and then functionalize the surface of this nanoparticle. The nanoparticle is radiolabeled for nuclear imaging with alendronate. Alendronate is a molecule that is a biphosphonate that has really good affinity towards calcium. So doing this functionalization, we can drive our nanomaterial towards microcalcification. Here is an example between the current gold standard for atherosclerosis preclinically, that is an optical Prof, and our particles. As you can see, the uptake profile is very similar. But here now we have a main advantages. Now we can do in vivo imaging, something that we cannot do with the optical imaging. And we can also quantify how much of our Prof is in the atherosclerosis place with the progression of the disease. So this is also like a very relevant advance. So with this, I hope I have convinced you that nanomaterials are really good to boost the field of medical imaging. But this is a two-way transfer of knowledge. So we can also use medical imaging to boost the nanomedicine field. And how is that? Because with medical imaging, we can study in a very easy manner the pharmacokinetics and biodistribution of nanomedicines, something that is usually very difficult to do by any other analytical techniques. So in this first example, we developed a nanomedicine for the intracellular delivery of monoclonal antibodies. So in this case, what we did was to take the advantage of these antibodies to radiolabel them and to do the following by PET-CT imaging. This is the biodistribution of the free antibody. This is the biodistribution of the antibody conjugated with the nanomedicine. So here what we are actually seeing is the profile of the nanoparticles after administration. We have the normal profile, elimination organs, liver and spleen. We found a small surprise with the mesenteric lymph nodes. It's not important for this talk. But what is important is that we can quantify very well where our nanoparticles are. We can calculate how long our nanoparticles are circulating in a very precise manner. In another example, this is actually was the first work I started to collaborate with Anna Roche and with Martí back in my time at King's College. We studied the biodistribution of two silica microparticles with the same apparent size, 1 micron each, but with different shapes. One is a rod, one is a sphere. We wanted to see if is there any shape-dependent biodistribution differences. As you can see in the images, they are, of course, and we can quantify them as well. This is the retention in blood, and of course, we can quantify how much or how many particles we have per tissue, which is very relevant if we want to exploit nanomedicine further. So again, I hope I have convinced you that we can boost nanomaterials using medical imaging. And as a last example, I'm going to show you something a little bit different. After many years of looking at images, biodistribution of particles by nuclear imaging, I started to wonder. So we are using a high precise tool that is nanomedicine, and I wonder if we are doing it in a precise way. Something that we cannot control or we do not currently control is the amount of particles we are administrating. We work on doses. I inject at 1 milligram per milliliter, 2 milligram per milliliter. These are millions, billions of particles, where 90% of these particles go to liver and spleen, and then the other 10 or so, if we are lucky, go to the place where we want. So I started to develop a method for the precise administration of particles. That took me a while, but finally, we achieved to inject a precise number of particles from a more conventional dose of 1 million of particles. We are usually working millions, billions, okay? But what happened when we start to decrease the doses of particles? We start to see these hot spots here in the lung that we characterize them as distal neutrophils. That's something we didn't know. We didn't know just because we didn't have the methodologies to inject a precise number of particles. And maybe we are doing it with many other formulations and we are not aware of that. So I just try to be precise with a precise tool. As you can see, we can inject only one particle. There is nothing more precise than that, just one particle. So we leverage this, and having the knowledge that all the radioactivity in this case is concentrated only in one particle, we can track how the particle moves inside the body. In something that we have called a biological GPS, we can see how the particle travels with the time inside the mouse, and we can relate that with the blood flow dynamics of the animal. So in this case, the particle is traveling through the lung capillaries. We can calculate the dynamics of the lung capillaries, something that is not possible to do by any other imaging technology currently. So I think this is quite relevant, and we will try to fully exploit this technique here, which comes to the next slide. What is going on? What is going to happen in the short term? So we are going to exploit this single particle tracking stuff. We will exploit it first, or more or less at the same time, but first we are trying to do it in cardiovascular diseases. Then we will need much better single particle tracers or more complicated ones to study blood flow dynamics and cell migration in the tumor microenvironment, something that is quite challenging. We are also developing methods to obtain the same imaging performance with few hundred particles that we have now with millions and billions of particles, and try to see if we can be more precise. And we will combine all this knowledge to develop precise nanocarriers loaded with RNA for the immunomodulation and lung repair in pneumonia. All of these have the same goal, that is to use real-time quantitative positron imaging with nanomaterials to obtain new biological information that we cannot see right now, and try to develop more precise therapies, increasing at the same time the precision with we are doing this. And that's it for me. I would like to thank all the people in the nanoparticles and nanocomposites group for the big welcoming I have had in the last year and a half. People who is dealing with me on a day-to-day basis, which I think is not particularly easy. The collaborators, and I would like to take a moment as well to acknowledge and thank the fantastic services we have here at ICMAB, because they have helped me a lot over the last year and a half. And that's it. I think congratulations are in order. 40 years is a big, big milestone. No pressure for early career researchers. We only have to do few decades of excellent research. So no pressure. And thank you very much for your attention.
Thank you very much, Juan. The floor is open for questions.
Juan is a visionary because he sees that we'll have a new building.
You see, it's not that difficult.
Besides that, I have a question. So you comment on the advantage of nanoparticles to increase the retention, but
Sorry?
You increase the retention.
The retention, okay.
No, compared to molecules.
Yes, the circulation, you mean.
Or the
retention in tissues, or
no, yeah, the time it is available for doing what you want.
Okay, yeah.
But what about the clearance? Is this a problem, or what is the balance between this? Because I suppose the clearance is lower as well.
The honest answer to this, I think, depends to whom you ask. My point is, when we talk with clinicians, which I think is very important, they are, of course, a little bit concerned about the long-term biodistribution of the particles, okay? Even though we are using biocompatible materials and nothing should happen, they prefer something that you inject, that act quick, and is fast eliminated from the body. Here with particles, depending on the composition, we can have the particles in the body for weeks to months, okay? The point is, all the clinical studies that have been done so far, I think the first one was with liposomes back in 2005. So we are not that long in experience with clinical studies, right? None of them have shown any kind of long-term issues after the administration. So my point will be that I think the advantages, the benefits we have of using nanomaterials overcome the limitations of having them for longer term in the body.
Thank you very much, Juan. I think that we are a little bit short of time, so we have to close. So thank you again.
So now it's a packed program. Now we are continuing with another outstanding researchers that we think that we want to follow the next years and deliver within the ICMAB. So it's Judit Morlá. He joined the ICMAB a year and a half after a successful postdoc in New York, in Mont Sinai. So she's a Ramón y Cajal researcher, and she will explain us about all her research. So the floor is yours. Thank you.
Okay, thank you, Anna, very much. I'm sorry that I don't have this nice picture as Juan, that you have to show me how you make that very beautiful. But of course, first of all, congratulations for the 40 years at ICMAB. And yeah, I think that the people make the center. So everyone that was here, especially from the beginning, pushing it, I think it's a big recognition. So yeah, here today I joined as a Ramón y Cajal just since February. So what I'm going to show you is just the research that I've been doing, mostly related to the interface on oncology, nanomaterials applied to cancer. So yes, well, if I'm doing it, yeah. So I think that the healthcare challenge here is quite clear. Around 20 million of new cancer cases were diagnosed worldwide in 2022. What it's about 1 in 5 people will develop cancer during their lifetime. So I think that there's no need to say much more about the challenge here. Just one important point is that the incidence here in this table, these are divided in incidence and mortality. And as you can see, some of the most incident cancers are also the ones with the highest mortality. And as estimated by the World Health Organization, this is expected to get even worse during the following years. No? And they justify or they estimate that this increase on cancer cases will be mostly related to, I don't know how to go back. Yeah, mostly related to pollution, high urbanization, lifestyle, aging. Also, we are living more years. No? So all of that, there are 2 main ways to tackle this. Either early diagnosis, that it has been widely demonstrated that as early as is diagnosed a cancer, higher is the survival, and this is transversal for most of the cancer types. And then the other way is treatment. No? We've seen different new therapeutics, different strategies to treat cancer, and especially immunotherapy being one of the most with the highest potential. So here I would like to show you several ways in which nanomaterials can help the diagnosis of cancer or can help in other aspects of cancer. So first, I will start with the diagnostic ones. And here, I would like to show you one example based on Raman. That was a Raman spectroscopy that we use here, nanomaterials to enhance the signal, to create surface-enhanced Raman spectroscopy. So this is the birational fingerprint of a molecule. And by obtaining it in these hot spots of these silver colloid hotspots, we achieve this much enhancement, this higher signal for the cell. So in a very simplified way, okay? But you have to imagine that this is like the fingerprint of this molecule. So tiny changes on this DNA or RNA sequences, we will see them here because it gives very high sensitivity. So based on that, we design a kind of a chip in which you combine these microRNA or DNA sequences And then by modifying the chemical surface of the particles, you obtain these controlled aggregation clusters in which you have your DNA or microRNA of interest in the middle. And this allows to identify, for example, in that case, adenine, like methylations in the adenine nucleobase, which is quite well established that it's a central epitranscriptomic regulator. So what you can see in here, I can go directly. So it's in the case of the single or even double-stranded, these slight changes in just one nucleobase we can identify through SERS. And in an even more clinically relevant case, it was the case of KRAS oncogen that you might have heard about it. But KRAS, it's one of the most popular oncogens for pancreatic cancer, also colon cancer. And so in here, combining this technique also with a statistical analysis, we were able to classify point mutations in this whole gene by partial least square analysis, combining the same technique that I was saying before, no? So for the cancer diagnostics. So that was just an example of how the design of these nanoparticles really helped to identify and classify different point mutations in cancer. And when cancer is diagnosed, and especially for solid tumors, surgery remains one of the cornerstone treatments. So about 80% of cancer patients are expected to need surgery at some point. And a concept very relevant and very important here is the lack of a complete resection during surgery, the fact of leaving there some cancer cells are highly related to a higher recurrence rate, which at the end, it leads to a lower survival. So here it's reported for ovarian, but it is quite established, again, for many other cancers. So nowadays in clinics, there's established the image-guided surgery, that it's a technique that helps the surgeon while is performing the surgery to contrast, to distinguish between the two tissues, no? The healthy one and the tumoral one. That's easy to be done in terms of that can be installed in the operation room. It allows high sensitivity, low cost in comparison to other imaging techniques. As we've seen, Juan, that would be amazing to have that resolution, but here wouldn't be the case. But in that specific case, which as I was saying, no, it's already included in the clinic, the main problem now, or more than problem, it's a limitation, it's the need of contrastation. So we need contrastation fulfilling all these clinical needs of biocompatibility, brightness, photostability, and so on. So in here, what we have worked, it's on designing nanoparticles, now following the same Miller idea here, designing fluorescent nanoparticles so that we can load many fluorophores in this small volume of a nanovesicle. So in here, we exploited quadsomes that are small unilamellar vesicles of about 100 nanometers composed by a surfactant and a cholesterol, forming this synton. And those quadsomes can be designed to be multifunctional, and they are prepared by the Delos manufacturing process, which is an eco-efficient process since it uses CO2, compressed CO2 as a solvent, and it allows a high control of the molecular assembly, so which leads to highly homogeneous nanovesicles in terms of size, lamellarity, shape, and also very important thinking on clinical translation, no? The scalability of this process, and it's also GMP compatible. So thinking on the final application, that's also important for the preparation of those nanovesicles. So exploiting this, what we've done is to incorporate fluorophores. In the past, we've done other fluorophores in here. Today, I'm going to show you the Fred pair example. So in here, we included a donor and an acceptor, Fred pair, and the idea was that by having this larger, this larger, I don't know how to say it now, this larger from the emission to the excitation window, that it's an advantage for the readout, for having less scattering during the imaging. And in addition to that, it has many other advantages for following also the stability of the particles. So here there is the design of 4 different systems and the cryo-TEM of about 110 nanometers in that case, and the storm images what you see in black inside, no? And all of them were showing this high homogeneity vesicle to vesicle. And most importantly, in terms of photophysical properties, we were reading here the Fred efficiency once normalized to the donor, and you can see so the emission of this acceptor is having this variation depending on the loading of the nanovesicle, giving us this orange bar that it's the quadsome, what we call 81, with the highest Fred efficiency. And when we studied this particle to particle with turf maps, that was also very nice to see that the heterogeneity that we have. So depending on this loading, this also related the homogeneity, the heterogeneity in that case, for example, that this different loading gave us. No? So we obtained this optimal loading with the optimal Fred, and also a very outstanding brightness, which it was reported to be this 10-fold brighter than quantum dots, and quantum dots are a marketed product recognized for being very bright. And in addition to that, together with other data, stability, in vitro biocompatibility, for example, here, 5 months of physicochemical stability in terms of size, shape, and that, but also in terms of optical stability, you see that the Fred emission was preserved for at least 8 months. Then all this data together with other that we're not going to explore now, it was the foundation to transform this research product into a clinical product. No? So in here, through valorization projects, we have evaluated the possibility to exploit this in a clinical setting, and it is was have been working in collaboration with Germans Trias y Pujol, and now recently, it was last February, we filed a patent in it for this specific case. So yeah, for that. And just to conclude, I don't know if I have, okay, perfect. Just 1 minute to conclude. Also, as future steps, I just wanted to add that we've gone from diagnostics, the tumor resection, but also treatment. So in here, it's quite clear that nanomaterials can be a game changer, and that's the idea, especially for the tumor priming. So immunotherapy nowadays is a very promising technique, very promising treatment for cancer. However, there are a lot of tumors which are not responsive to immunotherapy, which they are called cold tumors, completely immunosuppressed. And this is a real challenge because into that tumors, which are also the most aggressive, pancreatic, colon cancer, they do not respond to immunotherapy. So one of the ideas here is to prime these tumors, meaning shifting this landscape from being non-responsive to responsive to immunotherapy. And this can be done specifically via 2 different ways. Now it's emerging the 4th one, that it's by diet and other stuff, but we will limit to 2 that would be physical, like radiation therapy, ultrasounds, or photodynamic therapy. That it's what I've been working during the last years at Mount Sinai. And this gives really good results, especially when you do it as a co-therapy or pharmacologically. So you can add a drug. No? And here it's also, again, where nanomedicine plays an important role by giving this reaching, the idea of to reach these nanomaterials into the tumor microenvironment to tune or to make this shift into a more responsive tumor, turn it from cold to hot, and then can be potentially treatable with immunotherapy. So with that, I would like to thank everyone that has been involved here, people from Nanomol Bio, also from other groups during this journey, funding, very important to be able to pursue all of that. And a special thanks to ICMAB, and congratulations, happy anniversary, these 40 years, and happy to be here to continue the journey at ICMAB. Thank you.
Thank you, Judith. So if there's any quick question, I think she will be around as well. So if there's any question? So I have a question regarding, just very briefly, how you see the potential of these new contrast agents and these quantsums really going to, have you actually seen that that the doctors are really open to accept it and they change? Because this is always difficult.
I think yes. I mean, I'm always very optimistic, that's right. But also, I say yes because there have been already examples of clinical trials with fluorescent nanoparticles that at some point didn't work for several reasons. But I feel, I mean, in here, for example, there's one clinician on board of that, and since the beginning, he was really fascinated on the idea, but also because it's a real need. So they are already working with contrast agents. So they know it's just the fact of having a clear biocompatibility and no toxicity effects. And so if you are able to control that, I really see the
good to have the doctors on board then. So let's join me to thank Judith again, and then we will continue with another keynote speaker. As you have seen over the last 2 days and also last 2 years ago, Ramón Martínez Máñez is one of our members of the Scientific Advisory Board. He has been helping us, and also is a professor at the University of Valencia. University of Valencia. He has been holding, he's working on drug delivery systems that we will present it, and biosensors. He has been holding also very different ranges of responsibilities. He was the director of CIBER-BVN. Now he is the director of the Nanobiosis unit. So we were very thankful for him to actually accompany us over this time at the ICMAB and being in our advisory board. So the floor is yours. Thank you very much.
So thanks a lot, and well, to me it's a pleasure to be in the Scientific Advisory Board, and well, and two things: congratulations for the anniversary, and congratulations also for the work that we are doing here. That is really impressive. So I would like just to show in half an hour some of the work that we develop in our groups related with nanoparticles, and also some proofs for biomedical application. And well, most of the work I would like to show you are based on getting materials. We're interested in that, and we are developing this concept for many years. So basically, a gating material is a material that contains a molecular gate, and that can be defined just as a system that can allow mass transport, and that can be open or closed on command using external stimuli. So the idea is quite simple: is to have a porous support, and you can load the different molecules in inside the pores, then you anchor on the external surface that molecular gate in order to have systems that show zero release, but that upon the application of a specific stimulus, the gate opens, allowing the delivery of the cargo. That is the idea of most of the systems that I would like to show you. So we work with mesoporous silicon nanoparticles, even though you can work with different porous systems. So we prepare nanoparticles of that size. This is about 100 nanometers. As you see, those white lines are the pores. So as I said, you can put inside the pores different molecules, then anchor on the external surface that molecular gate. You have nanoparticles with zero or nearly zero release, as we will see. And upon the application of a specific stimulus, the gate opens and now allow the delivery of the cargo. This is the idea of some of the systems I would like to show you. And we have used that for two main applications. One of them is for sensing, and the other one is for drug or cargo delivery. So just me show you some examples on sensing. So the idea is quite simple here. So that represents a porous support that is now loaded with a reporter, usually a fluorophore. Then we can anchor on the external surface a binding site that form a complex with a bulk molecule. So the system is capped until there is a molecule that has more affinity for the binding site. There is a displacement reaction and a delivery of the cargo. So as far as the delivery of the reporter is just induced by a unique kind of molecule, then we have a sensor. So we have done quite a lot of work on that. Just to show you some examples in the past, this is for the detection of mercury that will induce the delivery of the cargo. That other one that allows to the naked eye to see amines of a certain length. This is just a first example of using those nanoparticles for the detection of DNA from a bacteria. This is a more recent example for the detection of papillomavirus. So we are working with that concept. Let me show you just another example and explain a little bit more how it works. So this is a porous system. And then we anchor on this is loaded with rhodamine. This is the reporter. And then we anchor on the external surface a molecule that has a small oligonucleotide that coordinates with that one complementary sequence here. And we have in the middle of that larger DNA a sequence complementary to the gene of interest. In that case, we wanted to detect Pseudomonas aeruginosa. So the system is capped until there is the DNA of the target pathogen. In that case, there is a displacement reaction because of hybridation and delivery of the cargo. So we can put that, for instance, in lateral flow tests, in which we can see the delivery of the dye. And here we have a result with clinical samples. Those are clinical urine samples for negative samples. Those are patients that were infected with Pseudomonas. This is in collaboration with the Hospital La Fe in Valencia. You can detect that without doing any kind of manipulation to the sample in just some few minutes. So let me show you another one. We are interested also in that concept, into detect drug of abuse. This is one of very well known. This is the gamma hydroxybutyrate. And this is the system is the same. So we have a mesoporous silica nanoparticle loaded with rhodamine. In that case, we anchor on the external surface that molecule that is a Valkofen derivative. And then that has some interaction with that GABA receptor. That's a protein, it's quite big, so it caps the pores and inhibits the delivery of the cargo. But in the presence of the GHB that has more affinity to that GABA receptor, there is a displacement reaction and delivery of the rhodamine. So this is how those systems perform. Typically, this is the fluorescence in the medium, so how much rhodamine has been released to the medium as a function of time. If you add the nanoparticles to a buffer, there is a poor delivery of the cargo. And if you repeat experiments in the presence of, in that case, the drug, there is a clear delivery. And most of the systems, well, of course, they are selective. So that responds to the GHB drug, but not to other drugs. So this is for sensing. We have more examples, but let me to show you how we can also use those systems for cargo delivery, for drug delivery. We have done also quite a lot of work here. So we have examples in which we can deliver the different cargo or different drugs using a stimulus like light, changes in the temperature, but also alternating magnetic fields, redox reaction, changing the pH, or the presence of enzymes on some molecules and biomolecules. We will see some examples on those just in a moment. So those are examples that we started to do many years ago. This is one in which we can deliver the cargo using light, a change in the pH. These are called nanoparticles. This is another one. This is the first example that demonstrated that gating nanoparticles can be used to deliver a cargo in water using a simple change in the pH. This is another one for the delivery, just making a change in the temperature. And here there is another one. This is a first report in which you can use peptides to cap the pores and use some enzymes in order to release the cargo. So now we have developed quite a lot of systems on that, and we work mainly in those areas, so mainly in cancer therapy, mainly related with senescence. So this is a field that we are interested, but we are also working now in immunotherapy. We have done some work in inflammatory diseases, and we also use those nanoparticles for the elimination of bacteria and fungi. But I would like to show you, I cannot just show you all the examples, but I would like just to focus on this, how we can target senescent cells. So we are interested in senescent cells because, as you may know, this is accumulation of senescent cells. It's a hallmark of most human diseases. And what is more interesting is that there is some proof that elimination of senescent cells might provide therapeutic benefits. I'm very interested on that subject. So that represents senescent cells, but it doesn't matter. It can be any kind of cells that you want to eliminate. And then you have to focus in different parts. So you can focus on, for instance, which is the lysosomal activity. But you can also focus on the surfaceome changes. So if there are any receptor that is overexpressed in the cell that you want to eliminate. But also, you can just have a look to the secretome and see if there are molecules that are specifically delivered, but the kind of cell that you want to kill it. So let me show you some examples on this for targeting senescent cells. If we go to the lysosomal activity, this is very well known. So there is an overexpression of beta-galactosidase. This is an enzyme that breaks galactose-galactose bond. And it's, as I said, overexpressed in the lysosomes of senescent cells. So the accumulation, there is a high concentration of that enzyme in senescent cells at very low concentration in normal cells. So we designed that nanoparticle many years ago, and some variation on that. This is a mesoporous silica nanoparticle. In that case, it's loaded with doxorubicin, and it's capped using a galacto-oligosaccharide. So you put those nanoparticles in a buffer. This is the delivery profile. There is a poor delivery of the cargo. If we do the same experiment in the presence of beta-galactosidase that is able to hydrolyze those galactose-galactose bond, then in that case, there is a delivery of the doxorubicin. So we have applied, well, then we have a system that targets senescent cells. It's quite simple. So in normal cells, the nanoparticles will arrive to the lysosomes. There is a low concentration of beta-galactosidase, and therefore no delivery of the cargo, but in senescent cells, in the lysosomes, there is a high concentration of beta-galactosidase that will uncap the pores and deliver the cargo. So we have applied that for many, many different systems. For instance, for pulmonary fibrosis, elimination of senescent cells improve the pulmonary function. So this is also applied for the elimination of senescent cells in tumors with quite good results. This is melanoma. This is another example with triple negative breast cancer. This is another one in which we saw that the elimination of the endothelial senescent cells reduced cancer cell migration. And there is another one in which we also saw that the elimination of senescent cardiomyocytes reduced the cardiotoxicity related with doxorubicin. So as I said, you can also design systems for targeting other specific characteristics of senescent cells. So that one just target the overexpression of some senescent cells in the secretome of the metalloproteinase 3. So this is again a mesoporosilicon nanoparticle. It's loaded with senolytic that kills senescent cells and is capped with a peptide that is substrate of the metalloproteinase 3. So that one, the nanoparticles target a receptor that is overexpressed in some senescent cells, is the DPP4 receptor. So this is a mesoporosilicon nanoparticle loaded with a senolytic and is capped using a polyethylene glycol. And at the end of the polyethylene glycol, we have an antibody that targets the DPP4 receptor. And the polyethylene glycol is attached to the surface using a disulfide bond. So inside the cells, there is a high concentration of a reductor that is glutathione. Glutathione breaks the disulfide bond and induces the delivery of the cargo inside the cells, not outside the cells. Well, we can use also nanoparticles for detecting senescent cells. So this is a similar nanoparticle as we have seen before, but it's not loaded with doxorubicin, but it's loaded with a dye. So the idea is that when the nanoparticle arrives, in that case, to tumors that are senescent, the dye is released and that can be monitored in vivo. We are also very much interested in developing proofs. This is another example for the detection of senescent cells, but it's based not in nanoparticles, but in molecular proofs. So this is a 2-photon structure that allows to detect senescent cells in a model of melanoma in ex vivo. This is doesn't work. No, son los vídeos. Yeah, sorry. Okay. Okay, so this is a kind of Theranostics system. So this is a molecule that has no fluorescence. Beta-galactosidase in senescent cells just hydrolyze that bond, and that produces that molecule that is highly fluorescent, but it's also a photosensitizer. So if we irradiate the molecule with we can obtain single oxygen and kill senescent cells. So we have a system in which we can detect and at the same time kill senescent cells. This is a Theranostics system, and this is another example in which we were able to detect, which is the burden of senescent cells in animals, just making a simple measurement in the urine. We are interested not only in senescent cells, but we are developing also proof for other biomarkers. This is, for instance, for detection of MAO, that is an enzyme that is related with aging, and we tested that in young and old mice. And there is another example for the detection of a drug, scopolamine, using a molecular case and a displacement reaction, which are very quite good results. So we are not working only with mesoporous nanoparticle. We are also interested in that concept. So we are using now gold nanostars because, well, if you irradiate gold nanostar with light, there is that is well known, a high accumulation of electromagnetic field in the tips of the star. What is less known is that the accumulation of electromagnetic field has been reported that can break chemical bonds. So then we decide to use the gold nanostars and anchor that molecule that contains doxorubicin and is attached through different bonds to the star. So the idea is that irradiation with that, the accumulation of the electromagnetic field breaks that bond here and release the doxorubicin free. It's like a kind of drug. So the system works, and we have tested that even in vivo models. So we grow the tumor, we inject in the tumor the nanostars, that system. We irradiate and are able to deliver the doxorubicin. This is just a proof of concept with quite good results in terms of reduction of the tumor size compared with other controls. So this is not hyperthermia, it's just the release using light of doxorubicin. Well, let me just, in the second part of the talk, shift to a different area. We are interested very much in that concept of nanorobots or nanobots. How do you want to call them? So the idea is those fantastic new futuristic systems that will perform complex tasks in our body. Well, this is, of course, still science fiction, but we can start thinking which are the characteristics that those nanorobots or nanobots should have. And of course, they should be able to sense the environment. This is not very difficult, so stimuli-responsive nanoparticles do that. We have seen examples for drug delivery, but also for sensing. But for many applications, it will be also interesting that those nanorobots or nanobots should have capabilities of communication, so they should be able to communicate between them. And for some applications, probably they also should have some kind of autonomous movement. So that was the idea of design, let's say, more advanced nanoparticles for different applications. So let me show you just how we have been working in those two areas. In the first part of communication, we are interested in designing system nanoparticles, but in general materials that can communicate between them. Well, if you go to a book and you see what communication means, it is defined as the process of establishing a connection between 2 points for information exchange. So the basic idea of the communication is you have a sender that receives a stimulus, and is able to send a message across to a receiver that understands the message and gives finally an output. This is the main idea of the communication. And that allows, of course, to make cooperative networks, very complex behaviors. Just we can think how we have developed humanity just using the language, or more recently also using electromagnetic communication, electromagnetic ways of communicate. Well, this cannot apply, of course, in using nanoparticles. And some years ago, we start to see how natures communicate. Of course, that is very well known. To give you some examples, cells communicate exchanging chemicals, neurons propagate and process information using neurotransmitters, and insect, bacteria, many mammals using pheromones in order to communicate with members of the same species. So it's quite clear that nature uses chemical messengers in order to communicate between them. So the idea was that, so can we design nanoparticles that are able to talk to one another? So can we design a nanoparticle that receives a stimulus, send a chemical messenger to a second nanoparticle, and that gives finally an output? That is the idea in which we have been working in the last years. So we demonstrated that many years ago, in which we designed, this is a mesoporous silicon nanoparticle in which we apply a stimulus. The stimulus is able to open the gate and deliver a first chemical messenger. This is another molecule that goes to a second nanoparticle. The messenger is able to open the gate and deliver a second chemical messenger. That finally go a final nanoparticle. That molecule is able to open the gate and deliver finally the cargo in the third nanoparticle. That's just a proof of concept was a dye. So the idea here is that you can apply a stimulus in that first nanoparticle, and that induces the delivery of another nanoparticle that is far away from that first one, using that concept of communication, in that way, using two chemical messengers. So this is a cascade model of communication, and we have been thinking in different models of communication. Here is another one. This is an interactive model of communication. So we have a nanoparticle, an input, that is able to send a chemical messenger to a second nanoparticle that gives a feedback, another chemical messenger to the first one, and that induces an output that, as a proof of concept, was just the delivery of a dye. So this is an interactive model of communication at the same, so that nanoparticle is only able to deliver the dye after the input if there is another nanoparticle in the solution to which it can communicate. That was the idea. So this is another example. It's a circular model of communication, and I would like to show you that in order you to see how the system works, a particular system. So those are made with nanoparticles, are Janus nanoparticles you have here. They contain a mesoporous part, and then are attached to a smaller nanoparticle that is a gold nanoparticle. So all of them have in the gold part an enzyme, and the mesoporous part is loaded with something and is capped using different molecular gates. And the system that in particular works in the following way. So the input is lactose. Lactose is transformed by beta-galactosidase to give glucose and lactose. Galactose is the chemical messenger that comes here. It's transformed by galactose oxidase to give hydrogen peroxide. Hydrogen peroxide is able to open that molecular gate, that is a redox molecular gate, and induce the delivery of another chemical messenger that is a benzoate derivative that comes here. This is hydrolyzed to give benzoic acid, and benzoic acid produces a drop of the pH and then opens that gate that is a well-known pH gate, and it delivers a reductor, and the reductor comes here, it breaks a disulfide bond, and finally delivers the dye. So again, this system, we have the same concept. So that nanoparticle is only able to deliver the cargo after the input if there are two more nanoparticles in solution to which that nanoparticle can communicate in that way. So this is how the system performs. So this is the amount of dye, the ruthenium complex that is released as a function of time. If we add all 3 nanoparticles in water, there is no delivery of the cargo. But if we repeat experiments in the presence of lactose, in that case, there is that process of communication and delivery of the cargo, of the dye in that case. So those are different systems in which we can be working. All of them are abiotic. This is the cascade model of communication, the interactive model of communication, the circular model of communication. This is another example with GUPs. We are now starting using GUPs for communicating them. And this is another example that communicates GUPs with silicon nanoparticle, in that case for the detection of a toxin. So we are thinking, those are quite theoretical models, let's say, but we are thinking in some applications. And we believe that that particular cascade model of communication can be used for signal amplification. I would like to show you an example of that. So this is a very simple system. This is a mesoporous silicon nanoparticle that is loaded with a dye. And the system is capped, is also rounded by a peptide that is a substrate of the metalloproteinase 9. So the idea is quite simple. So the metalloproteinase 9 hydrolyzes the peptide and induces the delivery of the dye. If we go to the limit of detection and see which is the amplification factor, so how many dyes are released per molecule of metalloproteinase, in that case, we do that, using the lateral flow test and a phone and a smartphone, and we can see the delivery of the system. So that's quite easy to develop. Well, the idea is, I was saying that if we see how many dyes are released per molecule in the limit of detection, that has an amplification factor of 900. That is not very bad. But if we do the following, so we have the same nanoparticle here, in which is exactly the same, and now it's loaded with penicillin. So in the presence of the enzyme, penicillin is released, and this is a chemical messenger that connects with a second nanoparticle that contains the dye and is capped using a penicillin aptamer. So this is designed in such a way that when there is penicillin in the medium, the aptamer just displays, there is a displacement reaction, and then we release the cargo, the dye. So if we do that and we do exactly the same, we see which is amplification factor in the limit of detection. In that case, we see that we have an amplification factor of 43,000. So that means that we are able to deliver 43,000 dyes, molecules of dyes per molecule of the enzyme in that case. Well, we are still working in trying to put here another one in order to see what we can increase that. Well, the example before, we are connecting nanoparticles between them. We are also interested in connecting nanoparticles with cells. Here you have a couple of examples of a living system. This is an interactive model of communication between a nanoparticle and this is a yeast, a cell. And this is another one in which nanoparticle is a kind of nanotranslator that is able to connect to different kingdoms. So this is bacteria and this is fungi. Well, just two more examples. If you go to the nature not only communicates using chemical messengers, but especially cells that have another way to communicate between them. That is that one. It's a communication through interaction with receptors. So this is also very well known. And for instance, it is also very well known that tumor escape from the immune system is related with that disruption of the immune tumor cell synapses. And we were interested to know if we can design a simple nanoparticle that could restore that cell-cell communication in order to T cells attack again to the tumors. That was the idea. So, and when we designed a quite simple nanoparticle, it's again a mesoporous-silica nanoparticle that is attached to a gold nanoparticle. This is a Janus nanoparticle. And then we anchor in the gold phase PD-1 antibody, so that target T cells. And we put here just peptide. This is that pH lead peptide that is very well known because it intercalates in the membrane of cancer cells. So the idea was quite simple. The idea was to see if those nanoparticles could just bring together tumor cells to T cells, and as I said, in order to T cells to attack again to the cancer cells. So the system works in vitro. I will show you just some slides, but it also works in vivo. So this is a quite simple model. It's a metastatic model in which we have mice. We inject those cells. Those are melanoma cells that express luciferase. So if you do that, the cells go to the lungs and start to make a metastasis. And you can follow the metastasis because the luminescent is growing with time. It happens exactly the same for some control nanoparticles, but if we add the nanoparticle that was designed that is complete, let's say in that case there is a reduction of you can see also here in histological analysis, there is a clear reduction of the metastatic clusters. So we are working now with those systems just using some imaging in order to see how the nanoparticles can attach, for instance, to spheroids. This is a melanoma spheroid. And we are also working with that concept, trying to do tumor-on-chip systems in which we have the melanoma spheroids in green. We put in a microfluidic channel PBMCs that are in blue that attack the melanoma spheroid, and we can see how that develops because we see that dead cells in red. So you can see here that is growing how we eliminate the cancer cells when we have our nanoparticle in the medium compared with the same system as a control. And we're working on that and different combination of antibodies to attach cancer cells with senescent cells. So let me show you another example. In that case, there is another way in which we can restore communication between cancer cells and immune cells. And this, with that simple concept, is reprogram cancer cells in such a way that they become incompatible with the immune system. So we have, as a proof of concept, do the following. So we have here the tumor In that case, we use lipid nanoparticle that is loaded with a messenger RNA that expresses the protein H of the measles virus, el sarampión, sarampión, el virus del sarampión. So the idea is that that will express the protein on the surface of the cancer cells. The immune system will think that this is the virus and will attack the cancer cells. That was the idea. So the system works, of course, in vitro, but also in vivo. So those are the lipid nanoparticles. We took just two group of animals. One of them were vaccinated against the measles virus, the other didn't. And only in those in which we injected the nanoparticles that were vaccinated, we saw that there was a reduction of the tumor size and, well, an increase in cytotoxic T lymphocytes that were infiltrated in the tumor. So that means that, in fact, the immune system was attacking the tumor. So this is the part of the communication. So we have developed systems in which nanoparticles can communicate between them, but also nanoparticles with cells. And we can also communicate, again, cancer cells with T cells using those concepts or restoration of the cell-cell interaction, but also making or trying to make cancer cells incompatible with the immune system. And finally, let me show you just how we can design nanoparticles that move and also for biological applications. So this is in the field of nanomotors. There are quite a lot of work in that area, mainly on micromotors. We're interested in nanomotors because applications, for instance, to treat tumors. We have done some work here. We're interested in chemical power sources using, as we will see, enzymatic reaction, but also intrinsic catalytic activity of some methods. And why? Because nanomotors are very good to enhance penetration through barriers. So that represents, for instance, a biofilm for a tumor. You know there is a poor penetration many times of the drugs. You can improve that a bit. Sometimes you encapsulate that in some nanoparticles. But if you do the same and the nanoparticles have an active movement, you can cross barriers that otherwise you wouldn't be able. So we have applied that, for instance, you can see here for the elimination of biofilms, and more recently also for the elimination of biofilms with an endodontic treatment. But I would like to show you the example for tumors, just one example. So we can use those nanoparticles to eliminate tumors. So those are Janus nanoparticles. We have a mesoporous part that you have here, mesoporous silica, that in that case is attached to a platinum nanoparticle. Okay, the system is loaded with oxorubicin. We want to kill cancer cells and is capped using a glucose oxidase. So that cap the pores, as we will see, and the system works in the following way. So the fuel is glucose. Glucose is transformed by glucose oxidase to give hydrogen peroxide, and that is decomposed by platinum to give oxygen and water. And as far as we will see that there is a chemical reaction in a part of the nanoparticle that induces a movement. Okay, that was the idea. So the nanoparticles will penetrate more in the tumor. That was the idea because of the movement, and eventually they will be internalized by cells. They will go to the lysosomes. The lysosomes are full of proteases. The proteases will hydrolyze the capping unit, so the glucose oxidase, and that will induce the release of the doxorubicin inside the cells. So at the same time, we remove glucose from the tumor. That's good because we don't want that the tumor has very much glucose to develop. We produce hydrogen peroxide, so some hydrogen peroxide and oxidant can also kill tumor cells. And we also produce at the end oxygen, so we can alleviate hypoxia, that is a common characteristic of tumors. So let me show you that, in fact, the nanoparticles move. Those are typical movement, this Brownian movement of the nanoparticles in the absence of glucose. But if the amount of glucose increases, you see how that is higher. There's a higher movement of the nanoparticles. That can be quantified just determining which is the diffusion coefficient. And you can see how the diffusion coefficient increases when the concentration of glucose increases. So this is the system. If you add the nanoparticles to a buffer, there is a port delivery of the cargo. If you repeat the same, but at a given time, you add the proteases that degrade the capping of the glucose oxidase. In that case, there is a clear delivery of the cargo of the doxorubicin. We have tested that in cells, a quite low concentration. There is an enhanced internalization in cells and also higher toxicity. We have tested that also in spheroids. So you can see those are slides of the spheroids. And you can see for a given depth how the nanomotors are more internalized in the center of the spheroid compared with a control that this is a similar nanoparticle that doesn't move. There is also improved penetration, but also improved toxicity at quite low concentration of the spheroids. We also tested in a tumor animal model. You see that there is a reduction of the tumor size when we use the nanomotor compared with controls. And finally, we did the same with organoids. Those are triple-negative breast cancer organoids in collaboration with a hospital in Valencia. And you see here how at quite low concentration of the nanomotors, we were able to eliminate the organoids. So, and that's all. So I hope I have convinced you that we can design nanoparticles that are able, well, to sense the environment. We have seen examples for drug delivery, but also for sensing, that we can design nanoparticles that can communicate between them using those chemical messengers, that we can also design nanoparticles that have autonomous movement and for some biological application. Let me to say that we work here in the Politècnica University of Valencia, inside that institute that is a universitary institute, but you also have a laboratory here in the Príncipe Felipe, in which we prepare here the nanoparticles and materials, and we test the nanoparticles and the materials in the lab we have in the Príncipe Felipe. We do their studies with cells and with animal models. And we also have a laboratory here, that is the Hospital La Fe in Valencia, it's the largest hospital in Valencia, in which we try to collaborate with clinicians also for several applications. Well, thanks to different support, regional, national, and international projects, and well, to my group, and thank you very much for your attention.
Thank you very much, Ramón, for this very nice presentation and all the future works. So is there any question?
Thank you. Very, very nice talk. My question is about how you produce or get the release of the UVs using this mesoporous silicon nanoparticle.
How I use the release of what?
The methodology to produce the UVs. You mentioned that a new application in your group or the next one could be using UVs, different biomimetic systems.
By UVs? What do you mean with UVs?
The UVs.
The groups, yeah.
Yeah,
okay.
the groups.
Yeah. No, we are working in groups, trying to communicate groups between them. Okay, we are not working yet in using groups for drug delivery. We have some ideas, but we have no results yet. We have just started maybe 1 or 2 years ago with that.
Thanks. I'm really interested in your talk. Maybe you show again about the nanosystem hybrid. I'm really interested. I'd like to say how to synthesis you convert the hybrid system. The other question is what is the difference about when you synthesis Janus nanoparticles? Because you have two phase. This is my question.
Yeah, related with the second, we prepare the mesoporous silicon nanoparticle. We use wax, Thera, and the nanoparticles just put inside the box, and they only give a phase, in that phase we then anchor chemically the gold or the platinum or any kind of nanoparticle that you want. And that works quite well. I mean, you have a very high percentage of the nanoparticles that are just that way. So one silicon nanoparticle, one smaller nanoparticle, for instance, gold or platinum. And sorry, the other question was, I didn't remember which nanoparticles you said that we want to see again? Or?
Janus, because I remember that when I was young, I synthesized the Janus, but I form a colloidosoma. I don't know, this is the same form. Yeah, this is a colloidosoma. Yeah, this is the question. Thanks.
Similar, yeah. Similar to that, yeah.
Okay, the last question.
Okay, thank you very much for the talk. I have a question regarding the doxorubicin release in the senescent cells, because the non-senescent cells also express the beta-galactosidase. If you leave them for more time or in a different quantity, also the non-senescent cells have the release of the doxorubicin in the system?
Well, if you do that, if you do that in vivo, there is a higher release in senescent cells. There is not zero release in, let's say, normal cells. But that can be several times more. So the question here is that you release a cytotoxic at low concentration. It's not toxic, but at high concentration it is. In vivo, in the models that we have used, they seem to work. To know what happened in vivo is more difficult always, but it seems that we target senescent cells and that we are able to remove senescent cells from different applications.
Okay, great. And another small question regarding the 3D models, the organoids, for example, I guess you use Matrigel as extracellular matrix.
Yeah.
Does the nano robots are able to enter into the matrix?
Yeah, they are. We are still working on that, but we have also labeled the nanomotors with a dye, and we can see how they arrive to the system, to the spheroid. Yeah.
Okay, thank you very much.
Okay, thank you very much to all. So Ramón will be around the whole day, so for sure he will be happy to answer all your questions. So please join me to thank him again.
Okay, thanks.